In the last video, I talked about cell communication between bacteria or unicellular organisms and a bit about multicellular organisms. But in this one, I want to talk about cell communication in multicellular organisms, including a little bit about development in those multicellular organisms. In plants and animals, you can trace development of a single organism, or you can trace the development of animals during evolution. That is, how has development changed from one species to another. So in green algae over here, you have unicellular things like Chlamydomonas. And then they're going a little further in Gonium here. The cells aggregate into a body in which most of the cells perform the same function, but it's safer to be together, and they also share resources. As the complexity of these aggregations grows, some of the cells become differentiated from the other cells, in that by the time you get to Eudarina and Pandorina, some of the cells are primarily photosynthetic, and other cells are structural, and then other cells actually have flagella, so they move the entire organism around. By the time you get to Volvox, it's even more specialized in that the inner colonies, these inner cells, eventually are released and become larger, with cells differentiating to look similar to the original organism. Eventually, you have much larger organisms where some cells specialize in reproduction, and other cells specialize in support. Other cells are primarily photosynthetic, and until you get to things like higher plants where they're vascularized also and can grow much larger. And so there's much more coordination among the cells in that case as well. So similar things happen in animals. In fact, some of the genes involved are similar. These are vertebrate animals. Here, just showing the development of those vertebrates. So if I showed you this top row at a certain stage, a very tiny stage, I don't think you'd be able to distinguish a lizard from a chicken from a human. But then later, you have these the genes turning on and off that will then create these differences among those organisms. So that all depends on cell-to-cell coordination, as well as intracellular signaling and differential gene expression in each one of those cells. Also, if you went before this stage that's shown at the top, you would definitely not be able to tell, unless you were an expert, the difference between these organisms. So early on, a large proportion of genes are turned on, and then there's sort of for vertebrates, there's a narrowing of the number and types of genes that are turned on, which determines that those organisms will become vertebrates. Now, if they were insects or something like that, then a different set of genes would define those body plans. So we'll talk a little bit about that a little later in this video. But first, if you go back to the very beginning, this is an animal development, you find that even in the egg cell, there's a differentiation between one part of the cell, one part of the oocyte, and another part. So it's called the animal pole and the vegetal pole. Early in development, there's a proliferation of these cells down here that will become the endoderm, and those will become internal organs. And there are these cells up here colored in blue on the surface, and they form very regular layers, and those are future ectoderm, which will become primarily things like epidermis and the lining of the gut and things like that, mainly things on the surface. Where the two meet, they'll form mesoderm, and these will be specialized areas in organs also, such as parts of the lung, parts of the liver, parts of the pancreas. A bit later in this process, there's an invagination to form a cavity inside of the organism, and that blastocele starts to shrink, and then you get in a gastrulation thing that looks like this. And so there's the ectoderm on the outside, the mesoderm, and the endoderm, which will become primarily internal tissues and organs. At this point also, where there's this gastrulation phase, some of the cells which interact with both the ectoderm and endoderm, part of the mesoderm are these cells called mesenchyme cells that tend to connect tissues together and cells together, but they also tend to move around and influence the development of other cells around them. So this is just a schematic showing some of those layers and what they become. So here's the ectoderm, which they become skin layers, pigment cells, neurons of the brain and spinal column, and then mesoderm become muscle, blood cells, and different parts of the kidney and pancreas, and then the endoderm, other parts of the pancreas, the thyroid, and lung tissue. And then there are the germ cells, the sperm and the egg over here. So those all come from different layers of embryonic tissue and can influence one another. In fact, these pigment cells those are one of the ones that can move around and are dangerous when you get a melanoma, because those kinds of cells normally move around, as I've shown in the previous slide. But when they're cancerous, you really don't want them moving around. But melanoma cells kind of go back to that embryonic pattern and start moving very, very early in the development of cancer. Blood cells are interesting in that they form so many different types of mature cells. So they start from these multipotential hemopoietic stem cells that reside in the bone marrow of certain bones. And then those divide and start to differentiate, depending on where they are and what signals they're getting from the outside, into either a common myeloid progenitor or a common lymphoid progenitor. So the myeloid progenitors can further develop based on the signals that they're receiving through their cell membranes, either a megakaryocyte that forms thrombocytes, things that help clotting, erythrocytes that carry oxygen, mast cells, and then a myeloblast. And the myeloblasts can further differentiate into basophils, neutrophils, eosinophils, or monocytes. And the monocytes can further differentiate into macrophages. And so these are part of the initial response to antigens, viruses, bacteria, etc., that invade the body. So responsible for the inflammation, for the fever, and for also secretion of fluids, sometimes that lead to pneumonia and things like that, but they're mainly there to try to help to limit and kill whatever antigens or invaders are in the area. And in addition, the macrophages can clean up essentially antigens that are left over, or they can actually chase bacteria and things like that that are trying to invade those areas of the tissue and cells. The lymphoid cells can then further develop into a natural killer T cell, which actually moves up to a cell that contains is presenting an antigen that it's captured, and it will actually kill or dissolve those cells almost immediately. It's a very quick response. These small lymphocytes then develop into B lymphocytes, which become some of which become plasma cells to secrete antibodies of a certain type, and then the T lymphocytes. And there's actually many types of T lymphocytes also, But these are really the central signalers and sensors in the immune system. So these are the ones that, in the case of HIV, they get infected, and so the immune system doesn't work. And these lymphoid cells also are responsible for long-term immunity. Development of the nematode has been studied in great detail because they can be grown on Petri plates easily in the lab. You can generate mutants, and the entire genome has also been determined also. They are also go through their developmental cycle fairly fast, and they're only about 1 to 1.1 millimeters in length, so they're fairly small, grow fast. And you can see here during development you get division of the zygote, and then further divisions, and finally development into the nematode itself. It's a fairly simple organism. There's a pharynx, a gut, an anus down here, and gonads in the middle. Now there are only two sexes. One is a hermaphrodite that has both eggs and sperm, and then there are males that have only sperm with no eggs. So two different sexes in this one. And it's about 1,000 to 1 ratio, hermaphrodites to males. So in most cases, the female just forms clones of herself over and over and over, lays eggs that develop into something that looks exactly like them. And it's only once in a while that they mate with a male to exchange more diversity in their genes. As I said, this organism has been studied in detail. This is a cell map of all the divisions that occur both in the embryo up here and then post-embryonic as the organism develops and starts moving around. So you can see in the end there are 959 somatic cells in the hermaphrodite, including 302 neurons, plus eggs and sperm are extra because they're always changing in number. But exactly that number of cells, and a lot of neurons actually, because they need the neurons to find food, to move things through their gut, and to find a mate and things like that. So, and the female needs to have neurons so that she can lay eggs. So the muscles for that have to be innervated also. But you can see a few places in here shown in red and blue and green. Those are places where during this pattern of development, some cells die, and they always die. So it's called programmed cell death, or apoptosis, in a special type of apoptosis. So how the cells divide, when they divide, where they are, is dependent on cell-to-cell communication. That is, what cell has just divided, what happens to each one of those cells, and what are they next to. So there are all sorts of signals, both genetic and cell-to-cell communications that are going on, as well as some hormonal, some long-term or long-range signaling as well. And in particular cell divisions, one cell divides off and goes on to develop into some other type of cell and divides, etc. And the other cell always dies. This is because some of the constituents of the cells need to be concentrated, and the only way to do that is to put them into one large cell, and then the other cell divides off and dies. And this happens in oogenesis also in animals, where during meiosis, only in the divisions, only one oocyte is produced, whereas in sperm, 4 sperma are produced during each meiosis. And that's mainly to make a large oocyte that contains a lot of ribosomes, a lot of other things, so that once that cell is fertilized, once that egg cell is fertilized, it can rapidly start dividing and start the early stages of development. Here's a more complex organism, Drosophila melanogaster, or fruit fly, which you can have mating of a male and a female in this case. There are no hermaphrodites in this case. And then you get a fertilized egg, zygote, you get initially nuclear division only, not cellular division, and then the cells start separating. The cell membranes develop around those, and you have cells, and then a patterning of segmentation occurs, and then you have hatching into a larva that looks sort of like a little slug. Those eat and eat and eat, developing all the time, and then there's pupation, goes into a little case that it builds, and metamorphosis, where the cells and tissues are reorganized around what are called imaginal discs. And so there's a total reorganization of the body into the adult, which hatches out, and then you get mating again to go around the circle. So there's the female's a little larger than the male, and there are the chromosome sets in each one. So they have a small number of chromosomes, but quite a few genes in the thousands of genes. Here's a little depiction of the life cycle of that one, going from egg, larva, gets larger and larger, pupation, and then hatching into the adult fly and back again. This is a fluorescence microscope picture showing this in one of these stages up here, where you can see some genes and some protein products are turned on in one area but not the other, and a different set of genes and proteins are expressed in a different area of the fly, setting up this segmentation pattern. And here's a life cycle pointing out a few of the details. So in the oocyte already, there is a differentiation in that cell from one end to the other, just as I showed in the other egg cells where you had an animal and a vegetal pole. This one, there are gradients of maternal gene products, mainly proteins. So then there's fertilization, and initially there are 9 nuclear divisions without cellular separations. And so this is called a syncytial blastoderm. And then the nuclei are then cordoned off or separated off into cells around the perimeter. There are a few cells at the end called the pole cells. Now those will become the gametes. Those don't go through any more cell divisions, or just a few cell divisions, so that there's no more mutation damage to those cells. So now we have a cellular blastoderm, and then you get expression of certain genes, so that they're called gap genes, which set up the initial patterning of one end versus the other end versus the middle of that organism. And often these patterns are produced by just a small number of genes and gene products. So you're getting fairly large differences from one end of the organism to the other, but caused by just a few genes and gene products that are signaling the cells to do a variety of things. The next set of genes and gene products to be expressed are pair rule genes. Now these turn on and turn off in different segments of that organism, different sets of cells. So they set up a patterning of those segments, one end of the segment versus the other. And then there are segment polarity genes, which further subdivide each one of these segments up here from the pair rule genes. And then once that general segmentation pattern is set up, then there's expression of the homeotic genes, which really affect the expression of many different genes down the line, that then set up the final body plan for that adult fly. Interestingly, these patterns are set up in Drosophila. This is Drosophila here. This is a mutant that doesn't do the same things. These patterns are set up in a similar way, but varying by either small or large amounts in mammals such as a mouse, a frog, there's a zebrafish, and then here's the fruit fly, a nematode, and then a sea urchin. So you see some of these same genes and gene products are being expressed in those organisms at different times during their development. But because there are these differences in when those genes and gene products are expressed, that affects the cells in those areas differently, so that then you come out with something fairly simple like a sea urchin, all the way to something much more complex like a frog or a mouse or a human. So here's a diagram of some of those genes and gene products that are turned on. So in the egg cell itself, there's a difference primarily in these genes. One's called CAD, one's called NOS, one's called BCD or bicoid, and then there's HB, which is called hunchback. And you can see the concentration of BCD and HB are higher in the anterior portion, and NOS and CAD are higher in the posterior end of the egg, so that there's already a difference from one end of the egg cell to the other as far as what's going to take place in the cells that eventually are in each one of those areas as the cell divides. And each one of these has an effect on the other. So NOS inhibits the expression of BCD, and BCD inhibits the transcription or expression of CAD. Then down here on the gap genes, these ones also affect genes down the line, such as hunchback, GT, KR, etc., to form a little more distinction in those now cells with nuclei. And then down here, multiple cells with nuclei. And then down here, there's further separation or segmentation because of the pair rule genes, and then more segmentation, more finer segmentation in the segment polarity genes. Then on top of that, all of this kind of feeds into producing a segmented animal that then can crawl around like a slug, and then you have these homeotic genes start to take effect, especially this early development, and then pupation, and finally the adult. And these are the homeotic genes. So a homeotic gene would not form a leg like this, but it would signal other genes, a bunch of other genes, to position a leg in that place. And they wouldn't form a wing, but they would signal other genes to form a wing in exactly that spot. And they have made mutants of these, and so you can make a mutant where you take one of the homeotic genes that's supposed to turn on and put a leg here, and you can put it up in the head region, and you'll have a leg coming out where the antennae should be. And there are some in the middle where you can form two sets of wings rather than one set of wings just by mutating one gene. So theoretically, you could do that in any organism by moving or mutating a gene to turn on in a certain place it's not used to turning on. And then you'll get an arm coming out where it's not supposed to come out, and a leg coming out in a place it's not supposed to come out, and so rearrange the organism. To develop a different organism, then these are turned on in slightly different places. So the blue lines up above are unique patterns for fruit fly, and then the red lines are unique patterns for wasp, and then the black lines are the same for both of them. So this is just in the egg, the oocyte. You can see there's already a difference. Some of the gene products are the same. There's CAD, NOS, hunchback, and bicoid is slightly different in that in fruit fly, it's not present in the posterior end, whereas in wasp, a similar gene product, OTD, is produced in a similar pattern in the anterior, but then it's very different in the posterior area. And then you have other genes later, gap gene expression, then becomes slightly different again in the Drosophila versus the wasp. And so eventually you get the two organisms that are similar in some ways, but very different ultimately in what they look like. One actually has a stinger, right, the wasp, and looks a little more slim in its body plan, etc. So there are differences in those, and just by getting slight differences in the expression of those early genes causes quite a bit of difference in the mature animal. Homeotic genes can be found in animals and plants, although the homeotic genes in animals are called Hox genes and the ones in plants are called something else. But they have a special pattern in that in the gene they have a variable region and at the beginning, a short variable region at the end, and then a homeobox. And that creates a homeodomain in the protein. And in Hox genes, these have 3 helices that are fairly well conserved. So these one-letter codes are for the each of the amino acids in those areas. And if you look at how those act, the part that's the variable region up here acts as a transcription activation domain for different genes. And then the homeodomain is the part that can attach to the DNA. So it's a DNA binding protein. So this helix 3 has a binding portion that binds in the large groove of the DNA. And these other two, helix 1 and helix 2, act as transcription factors to help to bind the RNA polymerase to the DNA for transcription. So these activate usually a large number of different proteins for specific functions. That is, these would bind to all of the genes responsible for making a leg or making an antenna to turn those on at a particular point in time in a particular place on the organism. And this is all done primarily with cell-to-cell interactions to tell each cell where it is and what it's just about to do. Hox genes in animals are well conserved, and they're not only conserved in sequence, but they're conserved in the same order along the chromosomes. So they actually turn on in those in that way. So number 1 is turned on, number 2 is turned on, number 3 is turned on in the head region of the organism. And then in the thorax or the neck region, these are turned on 5, 6, and 7. And then you have abdomen, arms, and legs turned on over here with 9 through 15, if they happen to have 15. Not all organisms have 15. For instance, humans have only 13, and 14 and 15 are missing now. But well-conserved group. You can see in fruit fly and many organisms down here, they only have 1 locus. But in vertebrates, there's been a genome duplication event, actually 2 genome duplication events here. So now there are 4 loci for those Hox genes, 4 loci for mice, birds, and amphibians and humans and things like that. But there are 7 or 8 versions in bony fish, and that's because there's been another complete genome duplication in those. So now starting with some of these, they had 8 loci, but they've lost 1 in some cases. And so there's only 7 loci for those. And you'll notice some of the genes are present in some of the genetic loci, and other genes are missing from certain genetic loci during evolution that's occurred. And that really has made some major differences in the structure and morphology of each one of those organisms. If you look at the evolution of these genes, then in its first column, that's the homeodomain, whether it has a homeodomain or not at all. You don't find it in unicellular organisms. But once you start getting multicellular organisms, you have more and more of these, and they control the one end of the organism from the other end of the organism. So in things like sponges and jellyfish, there's not a lot of differentiation in those between the head and the tail end. Then finally you get up to worms, where you have more differentiation from a head to a tail end. And then in arthropods, you have more differentiation and more genes involved, and more versions of genes. And finally working all the way up to mammals, where you have more changes than that. These last two columns indicate how many loci there are, how many genetic loci. So there's one down here, up to about 7 up here for these bony fish. And the number of genes is listed at the end, so that you have anywhere from one of those genes in sponges all the way up to almost 50 genes in, again, bony fish, 39 in birds and mammals, including us. Over here I've indicated the type of multicellularity, so you get more increasing cell type diversities as you go up into higher and higher regions on this phylogram. Here's a sort of rudimentary phylogram showing some of those genes and how they are turned on in different organisms. So in sea anemones and jellyfish, things like that, there's these red ones that are turned on in sort of the head region, and you can see a cross section of that where they're turned on to infold what will become the mouth and the gut. And then there's this main body region, and then a part down below that is the holdfast. It's a little more elaborate in nematodes, where you get differentiation of even more types of these homeotic genes, and then into arthropods, where it's more elaborate, and then into these chordates, including us. And here you have a pattern that is sort of down, which will eventually become the vertebrates, the vertebrae down here, a patterning or sectioning in this direction rather than across the body like you have in arthropods. But it's a similar pattern of turning on and turning off. In humans and mammals and vertebrates in general, you then have several cell layers that exhibit, that express those homeotic genes or Hox genes in different ways. So then you can get layer upon layer of different patterns of genes, and those Hox genes again are signaling other genes to turn on, and some to turn off, actually. And sometimes I'm saying Hox genes when I really mean the Hox proteins from the Hox genes. So it's really the Hox proteins that are acting as transcription factors to turn on or sometimes to turn off particular genes. So here's one example of limb development in a mouse. It would be the same as in a human, but you have Hox D9 turning on in the limb bud, and then Hox 9, 10, 11 turning on a little later. And then as the limb bud begins to grow, then it's Hox 9 here, 10, 11, 12, and 13 turning on there. And this shows that Hox 9 is actually turning on in every case, and this is Hox 9D. And then Hox 10 is turning on in this region, and then sequentially Hox 11, 12, and 13. So if you have all of these turning on, then you get these fingers and toes, and if you have only some of them turning on, you get a forearm, upper arm, or shoulder, or hip, depending on the limb that you're discussing. And these are normally controlled by other elements, early enhancers, which control transcription, and global control regions, which also are for transcriptional control. So their effect on early expression goes this way. The early enhancer affects these genes more so than they do genes further down the line, and then the global control region controls these genes here and has more effect on these than these. And although these are turned on in sequence, that is, 1 is turned on first up here, and 3 is turned on there, 4, and turned on in different regions of the organism, right? So these would be in the head region, these would be either in the abdomen region or in the extremities, the hands and feet in this case, and that's what's shown up here. But note these little arrows down here. The transcription of each one of those genes is in the opposite direction, so it's a little more complex than just going in one direction or the other. And here you can see the effects of that on how the shoulder, upper arm, forearm, and wrist and hand are formed, in that the early enhancer has more of an effect on things up to the forearm, and the global control region has more of an effect on those genes that turn on in the wrist and the hand. When a group of organisms is examined, that is, ancient organisms versus ones that are more complex and more derived, what you find is these Hox genes or homeodomain genes were very simple in the beginning and turned on sequentially based on where they were on the chromosome. Later on, you had some local control transcription factors that then would control one region or another. And so you would have a sequential turning on or turning off of certain genes, but just at a local level. Eventually, these enhancer elements and global control regions evolved to control first the local control transcription elements, and then later control the entire locus. So intermediate in here you have some control by both, and then ultimately the local control sort of fades out in an evolutionary sense, and you have then the enhancer elements and the global control regions controlling the expression of all of those Hox genes or homeodomain genes. The enhancer elements and global control regions differ from just a transcription factor, which would be these local control elements. And those are regions that attract certain factors that then encourage transcription of those genes. So they attract transcription factors and factors that attract other transcription factors, and eventually the RNA polymerase to express those genes. There also are other genes, not only the Hox genes, that are controlling some of the processes in development of these different tissues and organs and appendages. This is just one example of that. Here's HOX13 here, and then there's something called GLY3 over here. So when you have completely normal HOX13 and GLY3, then you get a normal limb, and this is from a mouse again. Once you have a heterozygote of a mutant of HOX13, then the appendages don't form quite in the same way. And then when you have a full mutant of HOX13, it's a club, essentially, rather than fingers, a hand and fingers. Now, the GLY3 controls how many of those fingers there are, fingers or toes. So that here you're getting a thumb and 5 fingers, or thumb and 4 fingers. Here you're getting sort of a mixture. They're sort of fatter and they don't look quite the same. And here with a double mutant of GLY3, you're getting multiple bones and fingers in those feet and hands. So when you have the double mutant, that is, it's homozygous double mutant, then you get a club with many, many different bones in it, and it looks more like a fin, which you might find in a lobe-finned fish, might look more like this. Then as you increase the GLY3 production in the HOX13, then you move up towards this way to get more defined, fewer appendages, fewer fingers, but more defined fingers and toes. This diagram shows a bit of the evolution of each one of those appendages. So you have an ancestral chordate, so we're chordates, and we have a notochord, and it leads to the vertebrates here. And so there are 14 Hox genes, 1 through 14, and then there's a genome duplication. The entire genome is duplicated in an ancestral jawless fish. And then you have some of those genes mutating out in those two loci. There's another duplication in genome to form what will be the line that becomes fish and mammals and amphibians. And so in the sharks, chondrichthyes, you have this situation where there are 4, and furthermore, there's a low concentration of Hox D13, which makes more of those fingers or toes, whatever, those bones in the hand or fins in this case. As you work your way up, there's been a further duplication in these bony fish. And so there are, starting with 8 loci, and some of those, again, mutate out of existence or go extinct. And in fact, one of the loci in each case has disappeared. So this one's missing that second D, and this one's missing the second C locus in each case. Then you go further up on this, and there's only been those 2 duplications, not the third one that you see in the bony fish. And now you're getting higher levels also of D13 expressed. So you're getting fewer bones, and these are the lobe-finned fish then that have fewer bones. They're starting to have a fatter fin, and then a sort of a fin tissue on the end of that club hand almost. And then further development here to form these, which are becoming now reptiles and amphibians, where you have a few fingers and they're fully defined. So you don't have fins anymore, you have actually feet. And then there are further increases in the Hox D products, the Hox D proteins. And so then you're getting fewer fingers and toes on those organisms, including in birds that have usually only 3, and more definition in those fingers and toes. And that's primarily from changes in the Hox locus, and in this case, specifically the Hox D13 locus. So then when you compare where and when these genes are turned on in different parts of the body. So here's in neuronal tissues, in vertebrates, in tunicates, and arthropods back here. You can see that they're turned on in slightly different places and at slightly different levels in different parts of the neural cord. In hexapods, that is insects, you have some that are turned on for along almost the entire body, and then that forms those long abdomens in these, where you get shorter abdomen regions, and then these ones here in the tetrapods, these are then innervating the arms and the legs, and in fishes, the forefins and hind fins in those organisms. If you look at somatic tissue, so this is now somatic tissue, you find a similar pattern, so that in tetrapods like us, these would be the arms and the legs, and those would match up with the neurons here in the arms and the legs. And in fish, they're expressed a little higher on that organism. So these are the front fins and the rear fins. And going down to annelid worms, those then have less distinction along the body. So you get these long worm-like organisms due to the expression of those Hox genes. And then here are the arthropods, where you're getting a fairly similar pattern all the way from spiders and scorpions to crustaceans to insects. A similar pattern in those because those are more closely related, although there are some differences. And those differences then you can see in the morphologies of those organisms. And then in a sea anemone, they have fewer Hox genes, and so there are fewer parts, and this is essentially the mouth and gut, or the mouth and the gut and main body, and then the holdfast, the thing that holds it to the rocky surface. So here's an ancient relative of ours. It's also a chordate because it has a notochord, so this is a true chordate, one of the first chordates. It looks sort of like a little worm with a head on it. And this is really the body here. So you can see the testicle, ovary, esophagus, siphon, pharynx, brain, little tiny brain, stomach, heart, etc. And then this is the tail, which consists primarily of a notochord. Now in us, we do have a notochord that develops early in development, but then it's sort of pushed aside, and our spinal column forms along where the notochord was, or where the notochord is disappearing. But you can see how these genes here in the epidermis are turned on, and then this long tail to form that epidermis. Muscles here, mainly here, to turn on muscles to move that tail. And then the nerves are turned on primarily here, partly for the brain, but partly to innervate the muscles that are in the tail, and then the notochord itself in the head versus the tail. Mainly this is setting up the basic body plan for the entire organism. So although what we find in human tissues and mammals in general is much more complex than this, the general pattern that developed in the initial tunicates is still within us, although it's more elaborate now. So now I want to talk a little bit about plant development. A lot of it's been studied in Arabidopsis thaliana. It's a small little weedy-like thing that has a rosette of leaves at the bottom and then puts up a flowering stalk with its flower, has a four-parted flower, and then this is development of a normal embryo here, and this is the development of a mutant that has a mutant in a single gene that causes a gross malformation of the embryo. And again, that's a homeotic gene, but it's not a Hox gene. Hox genes are that's a specific name for the animal homeotic genes. So these plants here are just mutants, usually varying by one gene for different homeotic type genes, or genes that are developmental regulation genes. This is a representation of a general developmental pattern for a typical dicot. In the ovary of the flower, you would have something that looks like this. Now, in gymnosperms, it would look different than this, but this is just for primarily an angiosperm. You would have what's called a female gametophyte. So this is a haploid generation. So in us, all we have is sperm and egg. There is no multicellular haploid generation. But in plants, there's a haploid generation. So at one end, you have what are called antipodal cells. There are two polar nuclei in the center that do not have cell membranes around them. And then there are two synergid cells and an egg cell, and it's the egg cell that will become the embryo. So there's also a male gametophyte that includes two sperm nuclei. So one sperm nucleus fuses with the two polar nuclei in the center, initially forming a triploid cell. That actually starts the nucleus starts dividing and it becomes polyploid. So that tissue, which is the endosperm, becomes polyploid. The other sperm nucleus fuses with the egg cell, and that becomes the zygote, which will become the embryo. And almost immediately there is a asymmetric division. And then there are different genes that turn on in different regions, eventually forming the embryo with the cotyledons, the seed leaves essentially, the shoot apical meristem, abbreviated SAM, and then there's a hypocotyl, which is the initial stem, and then the radicle, which is the initial root, including a root cap in the seedling to protect the root itself. In each one of these regions, then, you have some specific homeodomain genes and developmentally controlling genes turning on and off in those regions. So very early in development, this gene called VUSH for Vuschel then starts to express certain products that form the shoot apical meristem in the plant. And VOX down here expresses other proteins and turns on other genes to form the root end. And so this sort of shows how those function to control different products, to control the shape and form of each one of those organs. So VOX will stimulate stem cells, that is, cells that start dividing in the root to push the root out. That's stimulated by other proteins here, Cleave40, which activates ACR. Now that will then signal VOX to stop transcribing, stop producing that product, and that'll slow root development. So there's a feedback mechanism in here to control root development. So it doesn't speed up or slow down. It proceeds at a rate that evolutionarily anyway has worked for that particular species. So for instance, if you mutate VOX, then you get this kind of hairy looking root structure which is very disorganized, whereas if you have a VOX like this, one root goes down and produces a nice, straight root that then bends around rocks and things like that. In the shoot apex, VUS also controls two different genes, clavata1 and clavata3, and those control the amount of cell division at the surface here, and it also controls cells around here too. And if clavata turns on too much, it will actually inhibit VUS, and so there's again a feedback mechanism to control the growth of that shoot apex. If there's uncontrolled growth, then these cells just start to divide uncontrollably, and you form sort of a tumor on top of that, and the plant eventually dies because it can't produce the tissue it needs to produce leaves and stems and things like that. So each one of these cell types then is communicating through different protein products, different chemicals, sometimes RNAs, to control the growth of the cells adjacent to it, or sometimes a little further away than just adjacent. So the shoot apex then is highly organized, and it's the cells right in the middle that sort of control the production of cells that lead to the rest of the plant. But they're also organized into layers, and so there are what are called histogenic layers, which are essentially genetic layers. The first layer, L1, is always just the epidermis, and it can be either one, sometimes two layers in thickness. The next one, L2, it can be several cell layers thick, and then L3 leads to cells that are in the middle of the plant, called the pith. And those proceed through the leaf also in this manner, so that you have a green layer on the sort of the outside, this wider layer from L3 on the inside of the leaf, and then the epidermis covers the whole thing. And you can actually follow these cell layers by looking at mutants. This was actually from my master's degree, where I had a variegated plant, an ivy plant, which was white in the center of the leaf and then green around the margins, called Goldheart. And then you could actually follow those layers all the way up back into the shoot apex by looking at which layers had green chloroplasts and which layers had no were colorless, essentially. There were chloroplasts in there, but they weren't green. So here's looking at the shoot apex itself. The shoot apical meristem, and LP1, LP2, and LP3 are leaf primordia. Those will eventually form into leaves. And so you can see this very regular layering in these top layers, and that's called the tunica layers, meaning very organized. So they only divide in one direction. They don't divide in the opposite direction. So they only divide leading to a layer of cells. And that first layer then is the epidermal layer. These are the histogenic layers. Layer 2 actually consists of more than one layer, and you can see here where it pushes up a new leaf primordium. It's not only dividing in one direction, it's dividing in the opposite direction also, so that it pushes up a new leaf primordium. So it's highly organized, and it's all dependent on what the cells in the middle are producing, what chemicals, what proteins are being produced here, and how those signals are coming over to here to essentially take off an inhibition of dividing in the opposite direction. So then now they not only divide to form distinct layers, but they also start dividing in the other direction to sort of produce a lump of tissue instead of just a distinct layer of cells. On the right lower, the D, you can see some of these areas. So there's an apical initial zone. Those are the cells that are essentially stem cells for the rest of the plant. And by stem, I mean those are embryonic type cells that divide. And so these provide all of the genetic components for all of the other tissues, because they divide off and divide and produce all of the cells around that. Then there's a central zone, which you can see that's a little bit cleared of the cells there are growing, and you're getting an interaction of those different protein products produced by the initial cells and the peripheral zone cells. And then in the peripheral zone, you're getting more cell division, because now that's adding to the body of the plant, including to start producing leaves. And down here in what's called the corpus, you're getting further growth of the cells. So the cells become quite large with large vacuoles down there. It's providing just which will be the central parts of the stems and the leaves. Here's the view of the shoot region of a different plant, showing the shoot apex in red in the middle, and some of the genes that it's producing, some of the gene products it's producing, the proteins, that are affecting cells around it. So it's essentially inhibiting any leaf primordia from forming right in the center. It doesn't want to form a leaf right in the center. Then that wears off as you go further and further out on the stem. So then a leaf primordium is eventually formed. So most of these protein products are inhibiting certain things from happening. Also, further down, there's another leaf. Further down, there's another leaf. And eventually those leaves produce other genes, gene products, proteins or hormones, including YAB and CAN, which then determine which side is the adaxial side of the leaf, which will be facing the sun eventually as the leaf unfolds, versus the abaxial side that'll have the stomata and guard cells on it for gas exchange. So again, these are all cell-to-cell communications. Sometimes it's cells touching other cells, but often it's cells producing certain protein products and eventually chemicals. Some are hormones, which will control whether cells proliferate or don't proliferate, or whether they differentiate into a particular type of cell, or they don't differentiate. If you look at the pattern of leaves formed, then this is the youngest leaf, there's the next one out, there's the next one out, this is the next one out, etc. Different species of plants have different patterns of the formation of their leaves. And this is based on the relative concentrations of each one of these gene products. So here are some examples. These are opposite leaves where there are two leaves opposite one another, and then the next set is opposite that, and next set is opposite that. Decussate is when two leaves are in one direction, the next two leaves are in a perpendicular direction, and the next ones are perpendicular to that. And then you can have tricussate whorled, where there are three leaves in equal position on the stem, and then the next three are down from that, and the next three are down from that. Alternate are putting one out on one side of the shoot apex, and further down there's the next one around the shoot apex, another one further down on the opposite side. Then there are some with a whorled pattern where there are several leaves at one level, several leaves at the next level, several leaves at the next level. And then these ones are Fibonacci spirals. So this is a Fibonacci spiral here where there's 1, 2, 3, 4, 5, 6, etc. And this is a Fibonacci alternate, slight variation on the spiral where you have these adjacent leaves being produced, not quite in a spiral, but on crossing lines here for the next one. And again, these are all based on what what gene products are being produced in each one of those areas to stimulate or more often to inhibit certain structures from forming in those areas. Here are some examples of those. These are opposite. This is decussate, two coming out this way, two coming out perpendicular to those, etc. These are alternate, one there, one there, one there. Tricussate whorled here. Here are simply whorled, many leaves at the same level coming out, and then a Fibonacci spiral and a Fibonacci alternate pattern. In addition to the formation of leaves and vegetative structures, there's the formation of flowers, which takes other types of genes and gene products to stimulate or repress those. So for vegetative growth, you have light effects, primarily red light, far red light, and blue light. Gibberellin affects hormones. Temperature can affect this, so that you can have warm temperatures will cause one type of vegetative structure to form, and cold temperatures it'll change the vegetation, and nutrient effects also. And each one of these will affect whether the plant flowers or not, and they're centered around this particular gene, which is called leafy. So the thing is, when that is mutated, you never get flowers formed, you just get leaves after leaves after leaves. But if that's formed, then you can start generating a floral apex to form a flower or a series of flowers. And to form a flower, there are certain classes of genes and gene products that are involved. So there's this A type here, Apetala, and others that control both sepals and petals. And then there's a B type that primarily controls whether petals are formed. There's a C type, which affects petals, stamens, and carpels. And then a D type that primarily influences carpels, that is the female parts of the plant. And then an E type that affects many parts of the flower and is necessary for total flower formation. So just a bit about the morphology of flowers. So here's a standard flower with sepals down here. Often they're green and inconspicuous, and then you have the petals on top, which are usually colorful, but not always. Above that you have these stamens, which contain the anthers where the pollen is and the filament. And then finally the carpel, which consists of the stigma, the sticky part where the pollen sticks, the style here, and then the ovary down below with the ovules and egg cells inside. But not all flowers are quite the same, so here is a lily flower, and the stamen, the sepals and petals look somewhat the same. They're both colored, and they have 3 sepals and 3 petals, and then you can see the anthers sticking up, and then the stigma sticking up, part of the carpel. In C, this is an orchid flower, and they're quite different. So they do have appendages that look like petals, but they're actually tepals. So they're a sort of an ancient version of a sepal and petal that haven't differentiated yet into sepals and separate petals. So these are called tepals. And then way inside there are the anthers and the stigma. Here's a flower from an apple or a pear tree. And you can see the petals. You can't see the sepals. They're down behind and they're just green. And there's the anthers and the stigma. This is a different type of rose flower. This is a rose flower with the petals. The stigma or the sepals are behind. You can't see those. And then it has multiple anthers, many, many anthers, and a stigma in the center. This is a hibiscus with the petals around. Again, you can't see the sepals. They're back behind and green. And then it has a stigma, and then around that is a tube that has the anthers coming off of that. So quite a bit of diversity in flowers. And studies were conducted on different flowers to look at those class A, B, C, D, and E genes and proteins to see what effect they had. So here's a flower looking from the top. There's the carpel in the center with the ovules, then the ring of anthers or stamens, and then a ring of petals and a ring of sepals. And here, if you look at the normal condition, is what is occurring. So to make a sepal, you need the A genes and the A products have to be turned on. To get a petal, they take influences from the B gene and the A gene and their products to form a petal. To form a stamen, you need the effects of the B-type gene products and the C-type gene products. To make a carpel, it's primarily the C-type, except where you have the ovaries. And you also need the D genes and D products to be formed. And mutants were made of these. And so here's a mutant that doesn't have any A products. And in that case, you get a ring of carpels formed. And also within that, you have 2 rings of stamens and then a carpel in the center. If you get rid of the B products and B genes, you form a row of sepals, another row of sepals, and then 2 rows of carpels in the center of that. If the C genes are mutated, so you have no C product, then all you get are rows of sepals, and then petals, and more petals, and then more rows of sepals in the center. And finally, if you get rid of the E genes and the E gene products, all you get are leaves. No floral organs are formed whatsoever. So that's a little bit about cell communication and development in multicellular organisms, including a little bit about homeodomain genes, including the Hox genes, as well as some other developmental regulators, the genes and their gene products in animals and in plants. Okay, so that's it for this video. I will talk to you later.